everyone. It is noon, so we are going to get started because we have a lot to cover today. So welcome to the spring webinar for the Gabriella Miller Kids First Pediatric Research Program. My name is Valerie Cotton, and I'm the Kids First Program Manager. We are doing things a little bit backwards today since the program overview will actually be at the end of the webinar. So let me just kick us off by briefly introducing Kids First is a NIH Common Fund program focused on accelerating genetic discovery of childhood cancers and structural birth defects by building a data resource for the research community. We will kick today off with an update from the Kids First Data Resource Center, who will show you the latest features of the Data Resource Portal. Then we will hear from one of the Kids First cohort investigators, Dr. Wendy Chung, on the discoveries she's made with the Kids First Congenital Diaphragmatic Hernia project. And we will close with an overview and update of the program in the format of the frequently asked questions we received from many of you. We will open the floor to questions at specific points throughout the webinar. Please keep in mind that this is being recorded, and the recording will be shared on the NIH Common Fund website within a few weeks. So allow me to introduce our first speakers. In July of 2017, the Kids First program funded the Kids First Data Resource Center. The overall PI is Dr. Adam Resnick from the Children's Hospital of Philadelphia, which leads several different sites and their key roles as part of the Data Resource Center. You will also hear from Dr. Allison Heath, also from CHUP, and Dr. Vincent Ferretti from Sainte-Justine University Hospital in Montreal, who together will tell you about the progress they have made in developing the Data Resource Portal and associated workspaces. So with that, I will actually hand over the presentation to Allison. Okay,
thanks so much, Valerie. Welcome, everyone. This is Adam Resnick from the Children's Hospital of Philadelphia. It's a privilege for myself and the team to present to you our brief update regarding new features and development around the Kids First DRC portal. I'll start with a brief context for our effort and then turn it over to Vincent and Allison, who will demonstrate for you portal features. As Valerie mentioned, we always operate within a team, and all of us are also constantly online and on the portal. Soon you'll be able to actually interact with us within these environments. But today we'll actually focus on some of the search capabilities and use of the portal environment. While the three of us are presenting, we're representing really the work of a large number of people across multiple institutions who are making this possible and will continue to iterate on development and features of the portal. We all work across three different highly focused cores that really have diverse roles, but integrate to hopefully provide an environment and a platform that can really drive new discoveries on behalf of children. It's always good to start with our mission, which we constantly check against our activities, and our goal is really to support the alleviation of suffering through collaborative research and data sharing, building a platform that continuously iterates around connectivity and queryability and research between the pediatric cancer and development arenas for pediatrics. Much of our efforts focus around really key elements that together comprise much of our core functionalities. This includes a large-scale data that is being generated by EXO-1 investigators like Wendy, from whom you'll hear, the portal environment, which you get to see today, back-end resources that provide the appropriate access and security, the workspaces that Allison will demonstrate for doing actual research, all sitting in a cloud-based environment that promotes and supports non-local use and sharing of data. As context, I just wanted to provide how we think about building the portal, because we're still in the early stages of our activities. The portal launched just this past September, but really we have tried to align our activities and resources directly to the scientific method of supporting the observations, hypothesis, experimentation, analysis, and ultimate conclusions with impact that investigators have to harness in the age of big data and large-scale genomics across large number of diseases. Now, these activities can be very challenging. And really, we are driven by the unmet need that was really defined for our community and the Kids First program by Gabriella Miller herself. Gabriella, as a child suffering from diffuse intrinsic pontine gliomas, really promoted the acceleration of these processes through new technologies. And her frustration really was born out of a shared frustration amongst many investigators that are dedicating their lives towards meeting the amelioration of disease for these children. Gabriella was suffering from DIPG, a disease for which we yet have a cure, and was anxious to try and promote accelerated process. But she and many of us recognize that it's very challenging for individual investigators working in their own individual diseases to make the kind of progress in the absence of additional resources and platforms that can really harness data to its fullest extent. DIPG ends up actually being a great model for this, and Gabriella actually foresaw things that I think as a 10-year-old many of us are still learning about. For example, within DIPG, much research has defined new signaling mechanisms that involve genes like ACVR1. But at the same time that those researchers were focusing on diffuse intrinsic pontine gliomas, another rare disease community focusing on a completely different disease called fibrodysplasia ossificans progressiva, were also making discoveries on the exact same gene in children that did not suffer from cancer, but suffered from rare bone deformities and bone deposition. Similarly, at the exact same time that these activities were taking place, developmental biology researchers were doing drug screens in zebrafish to look for compounds that might affect the development of zebrafish themselves. And unbeknownst to them, we're defining the first compounds that ultimately could be used to treat both FOP and DIPG patients. However, this entire journey took more than 15 years to complete in ways that I think most of us hope we can change the course of looking forward on behalf of pediatric cancer patients and their community of partnered patients in the structural growth defect community. And so putting these together, our hope is that the platforms that we build and continue to iterate on develop a new model where together researchers across very different domains of research with varied expertise across different data modalities and research interests can come together on behalf of really driving an accelerated process of transforming data into structured information that can be queried, into knowledge that has meaning and purpose, and can be translated into clinical impact. And I think you'll see that what Wendy will present after our demo really is a great demonstration of these early efforts on behalf of our community. So with that brief intro, I'll turn it over to Vincent, who will begin the presentation and demo for the Kids First portal and some of the most recent deployments within the portal environment.
Thank you, Adam. So I'll share my screen. Can everyone see my screen now?
Yes, we can see it.
Yes, we can see it.
Okay, so thank you, Adam, again. So today I would like to focus my demonstration on the new tool that we have just released before the AACR meeting, at the AACR meeting, actually a few weeks ago. It's our new cohort builder. So you can see on the banner that it's still a beta version. We are aware of a few bugs that we still need to fix, and we will also want to implement some new enhancements based on user feedback that we received so far. So all this together, we will wait maybe a month or two before actually naming this tool with version 1. So the cohort builder is made of 3 sections, essentially. The first one at the top is the filter section. In the middle, you have the query section. And then at the bottom, you have the results section. So the results section offers a summary view, the one that you currently see on my screen, and a table view that I'll show you in a few minutes. So when no filters is selected, you see by default the entire kid search cohort. So you can see here that we have almost more than 8,300 participants with more than 34,000 data files for each of them. Here below you have a table, a breakdown of the number of data files per data types and experimental strategy. You have other breakdowns as well, for example, the number of participants per study, the number of participants per demographic characteristics, such as gender, ethnicity, race, and family composition, breakdown of by age of diagnosis, and here you have, for example, the most bar chart representing the most frequent diagnosis within your cohort, and at the bottom you have an overall survival plot for the participant for which we have actually applicable survival data. So, so let's start and do some query. So to help user to find what they're looking for, we group all the filters into a small number of intuitive categories like demographic, clinical, and quick filters are the ones that we think would be the most commonly used by the researchers. So for example, studies. You can see all the studies that we have now in kids first. So you have on the right side the count of the number of participants per study for each of them, and these counts are automatically refreshed as you add terms into your query. So let's select the first one, called the arts defect, and you select and you apply. And then you can see this term has been added in the query section. And then you can mouse over to see the complete term. Now, let's say I'm interested to do a trio analysis. So I will go to clinical, select only participant that would belong to a trio. So I go to family composition and select trio only, and then add a node. So you can see that the summary panel at the bottom is refreshing as you add terms. So now we have 888 participants who meet these criteria. So let's say I want to do some variant analysis. So I want to make sure that all the family members have variant counts, for example. So I can go here and select the family share available data types, and I can select, for example, GBCF. If I'm interested in more specific diagnosis for this participant, I can search for diagnosis here. So this is a text box. It's a very convenient way for searching filters. So I can type diagnosis, for example. I get the field diagnosis, open it, and then I can select, for example, ventricular defect. You can search. Oh, sorry. And let's say I want this diagnosis, ventricular septal defect first. I select it and I apply it. And then at the end, I have 9 participants who met these criteria. I can modify a query directly from it. So, for example, I'm interested to all the ventricular defects. So I can click them, and I can select one or more other defect, ventricular defect. I can select all, and then apply the filter. And then again, I get new result. So you can see first here that there's no survival plot. We don't have survival data for any of these 13 participants. So let me show you another query. So we can start a new query here. And this is a quite unique feature for the Kid Search Cohort Builder. So you can actually run multiple queries into a single session and combine them. So let me show you an example. let's say I'm interested in brain cancer, so I can type brain, find the study, apply it, and I want, then we can see we do have now a survival plot for these 953 participants. And if I want to be more specific, let's say I'm looking for medulloblastoma, I can apply this new filter, and then I get yet another survival plot for these. Okay, let me start, let's say I'm interested in outcomes, so I can start a new query again. Outcome, I do have a field called is outcome disease related. So I can click, I can say yes, select the yes value, so which means that the disease is related to the outcome for this patient. I can apply it. Then I get 162 participants, and now I can combine. So let's say I want query 2 and query 3, so I can select both and apply the AND operator between them. And then you see this is a very easy way to combine queries that you have previously built. So you see that now the queries are colored to help user to identify them. And you do have, well, it's actually executing this query, and then you have new summary, new result. You can always click back to a previous query to re-execute them and see again the results. So you can navigate across your queries, and you can see the change in the result. If you actually modify a query, let's say query 2, well, obviously it will affect the result of query 3 because of query 4, the fourth queries, because you see 2 and 3. So let's do it. You can remove one term, and you can see that actually this query has changed. You can even delete a query, a dependent query. You get a message, dependent queries will be deleted. Are you sure you want to delete? And you delete. And then you see that the query 3 now has only the term, involves only the second query. So let me show you another way to combine queries. So I'll start. Let's say I want target data, so I can search for target studies. Let's see this one. And now I'm interested at the age at diagnosis. So let's go there. Age at diagnosis. So this is another kind of facet. So you can select a range of values. So let's say I'm interested between 0 and 5 years old. And you can see now this bar chart shows only participants with newborns and 1 to 5 years old. Now you can duplicate a query. So if you want to modify it very slightly, so it's a faster way to build a new query. So you duplicate the last query. And then you can change the range, for example, directly from here. So you can, for example, 5 to 10 years. Okay, you apply. And now you can see that the bar chart is include only one column. And now you can do an OR, so you can select both. You do an OR. And then you see now that, as expected, you have more bar, the newborn until 10 years old. So this is the way you can work with this new tool. So again, the unique feature is the ability to actually do multiple queries, combine them, and now saving them. So for example, you can save a cohort. So once you're happy with this, you can save it, and you give a name. So let's say my first cohort. And you can note here that you are in this workspace, or the first cohort is now the title of your cohort is there. You can open the previously saved one. For example, you can open a new one, my second. This is a study that I had built before. You can open it, and this Okay. The table, so let's go back to the first one. Okay, so the table view now is basically the list of participants of these for this cohort for the current active queries, and from there you can download all the clinical data for these participants, the clinical data for the participants and their family members, and you can also download a spreadsheet for the biospecimen data associated to these participants. You can select columns, the columns you want to display or not in that table, and you can export this table in CSV or an Excel spreadsheet so that you can work with it with Excel. Now, let's go there. So the next step is to, let's see, I'm going here. I want to see the files that are associated with these participants. So I can click here, and I add the list. I open the first tool we released, which is the file repository. So I get all the files associated with these participants from my cohort. So I can apply for other filters. Let's say I'm interested only in RNA-seq, for example. And now I get all the RNA-seq for that cohort, files for that cohort. Now, the next step would be for me to push these files to our compute cloud environment, which is Cavatica. But you can see there's a lock here, so I don't have access to these files yet, because actually the system doesn't know yet if I have access, I'm allowed to access these files. So for that, I need to connect to the repository that owns these files. So for that, I go to the setting. And then you can see that actually the Kids First integrate integrating different data repository together, which are governed by different data access committees. So you need to connect to the good one. So we have two control access data sets, one governed by Kids First, and one and we have the NCI controlled access data. So target data is actually an NCI controlled data access, so I can connect to this using my ERA common account. So I can log in. Yes, I answered the question. And now the Kids First portal knows that I have access to some of the NCI controlled access data sets. And to know which one, I can click here, and I see that I have access to two studies, Target, acute myeloid leukemia, and neuroblastoma from Target. Now, if I go back to the file repository, I can see now that I don't have the locker anymore, but I have two icons. One that in theory allow me to download the data. So sometimes I'm allowed to download the data directly from the portal. For target data, we don't allow that. The only thing we allow is the ability to push to your workspace in Cavatica and for you to start for the user to start any analysis. So let's say I select some of them, about 20, and then I can push to Cavatica these files and start the analysis on them. So I did that. I will select the project, the Cavatica project I want to push my data in. So let's say I want to select this project. I have two projects in Cavatica now, two projects, Kids First Analysis and my project, and I copy these files to Cavatica. And I have success. So now Allison is going to show what we can do on Cavatica once we have pushed these files. So that's it for my demo. I just wanted to thank especially the Ontario Institute of Cancer Research and Christina Young team, who has contributed a lot to this new feature, the cohort builder. So I really they worked very hard to make this possible by the ACR meeting. I want to thank them especially. That's it. Thank you.
Great. Thank you, Vincent. I'll pick up where Vincent left off. I think you might need to mute. Okay. Great. Yeah, so we showed a lot. We've done a lot over the last basically 6 months from Kids First launch to now. the cohort builder, as well as you saw, some of this initial piloting of understanding how we can index and find pediatric data that's in different resources and provide a clear-cut way for both those different data sets to be properly governed, while being able, as a researcher, to really scientifically be able to integrate them and analyze them together. So we're really looking for a lot of feedback around that, and we're also trying to prioritize what some of our next steps might be around that. You know, as you saw, you know, Vincent showed some of the power of being able to search and find this in a space that's really geared towards pediatrics. So once you're on the Cavatica side, so Cavatica has been a good partnership with Seven Bridges and their platform, where we're also doing similar things. I'm not going to go through the full thing, but on Cavatica, you also do a similar login to Kids First Data Access, as well as the NCI Cancer Data Research Commons, specifically GDC data. And then once you do that, you land in a project like this. So we had set it up before. Me and Vincent are both in here. And you can really think of this as your local workspace in the cloud. So you really have control and governance over this project here. So you can invite members, remove members. You can bring your own data. You can bring your own workflows. And, you know, this is really a collaborative workspace that you can start analyzing the actual data, not just searching, finding, and downloading it. So, for example, if I wanted to add Adam here, I could add him. I could provide different types of permissions if I only want him to be able to read or execute. There's very fine-grained access control throughout the Cavatica and Seven Bridges platform. So I'll go and show you. So these are some of the files that Vincent was able to push over. And then once you have those files, what you want to do is bring different kinds of apps into that workspace. So you can do this locally. We've worked with a number of investigators to Dockerize and bring their pipelines onto the platform. And then we also have a number of publicly available apps. So, for example, if you're interested in DELI for structural variants, or you know, you want to look for the various different variant colors, deep variant, for example, you might be interested in running. And you can bring this into your workspace, configure it with different input files, and there's a lot of nice features that allow if runs get interrupted or things to be able to resume. So, for example, one of the apps we made public is the Kids First alignment workflow. So you can bring this into your own workspace, you can bring your own data, harmonize it with the Kids First data, and we've tried to make it as easy as possible so that when you first start this pipeline, we already have all the reference files loaded. You can copy that into your workflow, and really all you need to do then is select the input file and the name of your output that you would like this to be called. This one, and then from here, I can go ahead and run it. Oh, I didn't select my input. So I have to select an input, save selection. And then this is all API. There's a CLI as well for any kind of batch or large-scale operations that bioinformaticians might want to do. So this is going ahead and queuing it up, running it on the cloud, and you will get notifications as these different jobs finish. And so that's really good for these large-scale bioinformatics workflows, but increasingly we've been trying to roll out, together with Seven Bridges, different interactive features. So I'll just switch over here to a project where we've already preloaded a Jupyter notebook, for example. So this is interactive analysis here, data cruncher, and then Captain Crunch, because it's hard to come up with good names. And then this is already up and running in my workspace. I have different Python notebooks here where I can immediately have access to all those controlled access files in my project. And so I can write different code, I can do different filtering, create plots, share these notebooks with others. And now they've also rolled out a beta version of RStudio as well, if you prefer RStudio over Jupyter Notebooks. Other interactive features include a genome browser in place, so you can add some different files here if you want to take a look at the actual reads in place, and then you can go and actually interactively browse and view. There's a number of other different features for working with the data locally as well. So there's, a huge suite of that around this, and, we're really looking to find, feedback and understand, what's working, what's not working. Part of what we've been doing over the last couple months is rolling out a cloud credits pilot, where we have researchers providing different type of abstracts around Kids First data analysis. So far, we have 8 pilot applications that we've approved so far, and we are working on evaluating that results and understanding both what's accelerated on the cloud, what gaps there might be in features, and as well as different cost and sustainability models longer term. So that's really what I had for the Cavatica component. Anyone can log in and try it out. It's not just Kids First. So we are welcome to any kind of feedback. I'll turn it over to Adam to finish up our portion.
So thanks, Vincent and Allison. And again, this really represents a ton of work by a very large team across diverse institutions, working closely with the NIH and the different institutes and centers there. You'll learn more about the Kids First program and how the Data Resource Center fits within the landscape of sequencing centers and EXO-1's investigators as Valerie takes over at the end of the webinar. But I'll turn it over now to Wendy, who really, I think, serves as an ideal example of how investigators leverage the Kids First program and resources to drive and accelerate new discovery on behalf of children.
Great. Thanks, Adam.
I would like to introduce Wendy as well. We do pictures since we're doing this webinar style today. So our next speaker is Dr. Wendy Chung, who is a renowned and accomplished clinical and molecular geneticist at Columbia University. I don't have time to go over all of your accomplishments today, Wendy, but we do want to mention that she was one of the first researchers to participate in the Kids First program in leading the congenital diaphragmatic hernia project. So we're very excited to hear about your findings today. And I will turn you over as the presenter.
Thanks, Valerie. Let's see, this goes here. And hopefully this will go full screen in just a second. I'll go ahead and start talking while hopefully this loads up. And Valerie or someone, can you just confirm that people can see this?
I see some, oh, there it is, yes.
Okay, very good. So thanks everyone for having me today. I'm very glad to be able to tell you a little bit about what we've started learning about congenital diaphragmatic hernia. And let's see how this is. There we go. So for those of you who don't think about CDH or congenital diaphragmatic hernia all the time, this is a congenital anomaly that we see in terms of a defect in the diaphragm. The diaphragm is your breathing muscle, and it also separates or acts as a barrier between your abdomen and your what we call your thorax or your chest cavity. When babies have a defect in that diaphragm as they're growing in utero, what I'm showing in the figure on the right and that sort of pink stuff is that their intestines can actually move up into the chest and can compress the lungs and the heart and can cause problems in terms of lung development. And we see some of that postnatally. We see this congenital anomaly about 1 in 2,500 births. And unfortunately, it can result in a very high rate of mortality of about 30% because you can imagine it's difficult for a baby to breathe, and if this is not recognized early, can in fact cause those breathing problems. It can be associated with other things as well. Congenital heart disease is one of the things that we see most frequently, and I'm going to come back to that, but I think one of the real important things about the Gabriella Kids First program is the ability to look across birth defects, and that's one of the things I'm going to emphasize today as we're going through this. Let's see.
Wendy, I think your WebEx block is somewhat blocking the screen. If you could just pull it to the side.
Oh, I'm sorry. Thank you
for that. It's okay. It's not your fault.
Let me see how I can do this.
Try double-clicking it to make it smaller, and then you can grab it and pull it to the side. That's getting better.
Let me Maybe that's good enough. How's that?
There
Is
you
that
go.
better?
Good enough. Yes. Thank
okay,
you.
very good. Okay. So as we started looking at this now over a decade ago, there was some evidence that this might have a genetic component because we knew there were some children who had chromosome issues. We also knew of some rare genetic conditions with what we call single gene disorders, and there were not many, but cases of twins where we could see it running in twins or within siblings or even within families from parent to child. We also knew in some cases of animal models, in particular mice that had diaphragm defects in development. And let's see, hopefully my slides are advancing. Valerie, is it not just the down arrow? I know you gave me instructions that I've forgotten now. I've got it here now.
There you go. Yeah,
okay.
That's good.
Okay, very good. So as we did this, we put together a consortia of many different clinical sites around the country. You may see some of your local institutions. We've also since expanded, so we now have international sites around the world. And the point is that for relatively uncommon anomalies, we need to be able to put our heads together, and that's what we've done. As we've done this, we've had really wonderful families that we've been able to work with, and one of the things that we've done is not just studied them at birth or at the time they joined the study, but continued to follow them as the children have been growing up. And I have to say personally, as a pediatrician, that's been a great thing to do, is to watch the kiddos grow up. As we do this, we've taken many, many different data points or clinical information that we did about how they did in the neonatal period, how old they were when they went home, if they had other congenital anomalies, if they had other medical issues, if they were having trouble growing. One of the things we can see, as I said, is breathing problems. So we looked at that, as well as an associated condition called pulmonary hypertension. We actually had all of our echocardiograms read by a single center so that we could have consistency in assessing the level of pulmonary hypertension. And as we followed the children growing up, we've been following to see how they've been doing in terms of growth as well as development. So we've actually done some, I think, consistent, rigorous, standardized methods of assessing development at 2 and 5 years of age, which have been helpful for us. When we started this a long time ago, we actually started out by first using some blunter genetic tools out of a karyotype and chromosome microarray, and have gone on to exome, and then with Gabriella Kids First, gone on to genome sequencing as well. As we did this in the early days, as I said, there were some things that we could see by looking at kind of a high level. Chromosome conditions like trisomy 21 or Down syndrome, some translocations, as well as copy number variants. And just to give you a sense of what this looks like, they are distributed throughout the genome, so they weren't located in any one particular place. But I'm going to show you one circumstance, and I'm going to point out that we saw in particular a region on chromosome 8, 8p23.1, where we saw not just one individual with CDH, but actually a series of individuals with congenital diaphragmatic hernia that had regions that were all in 8p23, but they were varying in terms of size. And we could look at what we called a critical interval, the one that was shared by all of those individuals. And I'm showing down below the different genes that we saw within that interval that was deleted. And importantly, I'm going to draw your attention, I show here GATA4 as one of those genes that we saw within that interval. Again, it wasn't the majority of individuals with CDH. We certainly have not found one single gene for CDH that explains all of it. One of the bottom lines is that it's genetically complicated in the sense that there are many different ways at which we can disrupt diaphragm development. In the early days, after we were able to do that, we couldn't quite afford to be able to do genome sequencing, so we took a shortcut, not just we, but many others in the field. Your genome looks something like this. It looks a bit confusing and difficult to decipher, but if you know how it's organized, you can see some of these gold nuggets, as I'm showing here in terms of being able to see the Gettysburg Address of 7 years ago. And the point is that if you can focus on those gold nuggets of the so-called coding regions, or the portions of the genome that encode the proteins or the gene products that our cells make, then you can cut down the workload to about less than, not quite 1/100th, but a little bit more than 1/100th of the work that you would have to do. And so that was an important advancement in terms of our understanding. As we started doing this, one of the obvious things to do was to look at our families, where we saw more than one family member with diaphragmatic hernia, thinking that there might be a greater likelihood of something genetic if we saw it running in a family. So I'm showing you here one of our families, and colored in black are the individuals with diaphragmatic hernia. You can see the arrow in the bottom row is pointing to the child who actually was enrolled in our DREAM study. That child had an uncle, so his father's brother had passed away actually when he was just a little baby of diaphragmatic hernia, so that we knew that we had an uncle-nephew pair in terms of doing that. So we sequenced, in this case, the young man with the arrow, his mom and his dad, and started looking for what might be something related perhaps to diaphragmatic hernia. As we did that, we saw that he actually had a genetic variant, one that we call a missense genetic variant, or a single amino acid substitution, one single building block within a protein that was swapped out. Instead of a red block, it was a blue block. So it could be thought of as a somewhat subtle change. But I want to point out to you that that gene that was affected was that same GATA4 gene that I showed you back in the chromosomal region of 8p23. So before we were seeing that it was just deleted, that in several individuals we were missing that gene. Now we're seeing a more subtle type of change, again, something that would change the GATA4 perhaps so that the binding affinity for this transcription factor, or this gene that orchestrates and organizes the expression of many other genes, might be affected. So as we did this, again, we stopped and thought to ourselves, does it make sense that this might be the gene that's involved? And we thought logically it made sense. This was a gene that is expressed in the right time, in the right place. We knew it had been associated with congenital heart disease. We know mice, or at least some mice that had mutation in this gene, also had diaphragm defects. So it made sense to us. But we were starting to ask ourselves whether the specific change that we saw in amino acid 252, in fact, whether or not that was problematic. So we went back to our family and said, well, in fact, if it is, we would expect it to be traveling with diaphragmatic hernias in the family. And I've shown you here again, I've added the genetic results for the family. So what I mean to show here is anyone who has a minus sign is carrying the genetic change that we see in the GATA4 gene. And you can see that the young man, he has a minus sign. You also see his father also seems to carry the same change. And his grandfather also, we saw, carried the same change. We didn't have DNA on his uncle who had the CDH, or the diaphragmatic hernia, so we couldn't test him directly. But we did test other people in the family who didn't have any of the diaphragm defects, and they didn't have that change. But I remember being a little bit puzzled and saying, but I don't understand. His father seems to have this change. His grandfather seems to have this change, yet we don't see any diaphragm defects within them. How could this be? Maybe we have the wrong answer. Maybe we need to rethink this. Or maybe I thought we switched the samples. Maybe we accidentally, sequenced the wrong person, or made some mistake, or had some contamination. So we went back, we checked all of those things again, and then it sort of struck me one day as I was, believe it or not, waiting in a very long line and just sort of daydreaming, having my mind wander. And I thought to myself, well, maybe his father really does have a diaphragm defect and we just don't realize it. We haven't really looked very carefully. Maybe we need to go back and look. So we did that. We went back to his dad and to his grandfather, both. And these are MRIs, ways of being able to image and look very closely at the diaphragm. And again, they weren't complaining of any symptoms or any problems. But if you look at each place, I don't think you can see my arrow on the screen. But if you look at each place where the arrow is showing on the MRI, There are 4 of the arrows and the 4 different panes. Each one of those is actually a very small defect in the diaphragm where you can see things either bulging through just a little tiny bit, not enough to cause any problems with breathing, not any problems in terms of lung development, but clearly a problem in terms of how the diaphragm developed. And actually, since then, since we had this first realization, it's something that we've done repeatedly and realized that in some families, there are individuals who have very, very subtle changes that tell us that there is really a problem with diaphragm development, but thankfully hasn't been so severe that they needed to have surgery. So beyond that, we went to be able to see whether or not we might see genetic changes, but in a different family structure. So most of our families actually were not familial in the sense that there were 2 or more people with diaphragmatic hernias. Most of our families, there was only 1 individual with a diaphragm defect. And again, if you think about what I was talking about before, this is a really serious birth defect. If people didn't appreciate it, if you didn't get diagnosed quickly with this condition and make it to the right hospital with the right surgeons, really a generation ago, most individuals, most babies that were born with diaphragm hernias actually died from their condition. And when you think about that, if you think about if those individuals died, what happened with those genes? I mean, their genes died with them. How would they have been passed down in the families from generation to generation? It could have been as in the family I just showed you, but in many cases, they wouldn't have been passed down. So we thought to ourselves, maybe in terms of where these genes come from, maybe they're de novo mutations or new mutations in the families, and that's how they arise. So we started looking at that and looking at many of our families where we didn't have multiple people with diaphragmatic hernias. And we saw that about 6% of them had these copy number variants I alluded to before, and about 20% of them that had CDH plus something else. We could see what we thought to be a damaging or deleterious de novo genetic variant. And even the ones who had just an isolated CDH, although it was a lower percentage, it was only about 10%, about 10% of them had these de novo changes as well. So I'm going to do just a very brief primer because I know some of our families are watching today. So just so you understand some of the gobbledygook that we talk about. Having these de novo genetic changes is not specific to individuals with CDH. In fact, I can tell you I've done the experiments with my own 2 sons. We've looked in terms of my boys and looked at their genes compared to my husband and myself, and they literally have a little over 100 genetic changes that are different from either of us as their parents. They don't have diaphragm defects, and part of the reason is because they got lucky to a certain extent. Those genetic changes that happened anew happened to fall in places that didn't matter so much. One of the reasons is because I'm showing you here that for instance, this type of genetic change where you change this alphabet letter of a C to a U, it doesn't happen to change that amino acid in the protein. We call it silent because it doesn't change the amino acid. We all have many of those, and they don't cause us any problem. They don't change anything for our bodies. Some other changes are what we call missense. Changing this G to a C changes this genetic code for the amino acids, and it changes that genetic code from one of those building blocks called an alanine to another building block called a proline. Many of those changes are benign or not problematic. We have ways of being able to predict now better which ones will or won't be a problem, but many of those are just fine. Our bodies tolerate them without a problem. But there are a class of changes that, for the majority of individuals, cause problems. And again, it's the same type of change in a way. It's just a change of one alphabet letter of an A to a U. But in this particular case, it causes what we call a premature stop. Instead of making the full protein, it makes only part of the protein, and then it stops making the rest of it. And in many cases, in fact, probably the majority of cases, those disrupt the protein and how that works. So I gave you that lesson because as we've done this, we've looked at, from a statistical point of view, the frequency that we saw such changes in our individuals with CDH, and to be able to look at them by these different classes. So again, in doing these and then also looking at individuals without any of those congenital anomalies as a reference or as a comparison, we started looking at the frequency or the enrichment of that. So those silent changes that we talked about, we looked at different types of things. The black is showing all genes. The pink is showing a class of genes, about 2,000 of them that we call constrained, or they're kind of rigid. They like being just exactly the way they are. And then these in light blue here, other genes. And when we look at these silent changes, these ones that don't change the protein, we don't see really any difference. This dotted line here of 1 means that the ratio or the comparison is about 1 to 1. Again, no more, no less for individuals with CDH than for other individuals who don't have any congenital anomalies. On the other hand, if we look at the group as a whole of all of the missense variants, we see a little bit more. Statistically, these are error bars here. Statistically, it still is right near 1, so we don't statistically see a difference here. But now when we take the category of deleterious missense, or those that we have ways computationally to predict might be causing a problem, for those individuals with CDH, we see an enrichment or an increase in the frequency for all of those, either in constrained genes or other genes. And then in particular, if we look at the likely gene damaging, we see a much higher frequency, especially in this pink dot here for the genes that we call constrained genes. So we're starting to hone in on the signal. It's not telling us yet necessarily which gene within all of these is causing the problem, or which multiple of these we're going to be able to clear as CDH genes, but it's telling us that we're on the right trail. As we did this, one of the other ways, we looked at it in a couple other dimensions. We did have the sense, as I was alluding to before, that if you had not just diaphragmatic hernia, but might have another associated birth defects, something I've called a complex case as opposed to an isolated case, that there might be a greater likelihood of having a genetic or at least a single gene effect in those cases. And again, what I'm showing you here is the complex cases on the left of the slide in blue compared to the isolated cases in red. And again, you can see there's a higher frequency of those for the deleterious missense, and just slightly higher, I'd say really about the same for the likely gene damaging or disrupting variants as well. Now, interestingly, we were also looking at the differences by sex, whether or not there was going to be a similar frequency in boys or girls. We do know that CDH is slightly more common in males than it is in females, almost as if females are somehow protected in a way from this, or males more susceptible, depending on if you look at the glass half empty or half full. And so when we were looking again at these de novo variants, we saw in pink in the girls, we saw a higher frequency of these de novo deleterious missense variants or these likely gene disrupting variants, either one of those. It seemed as if for these girls, they needed to have what we think as one of these factors that was disrupting the development of the diaphragm that would have caused the problem for them. And the way I'm showing this is to be able to think about this as a threshold of liability, as one of the ways that we think about it, that, again, girls might be somehow protected. They have a higher threshold in this. Boys, for whatever reason, might have a lower threshold. And for these girls, it really takes a big sort of something pushing them hard to be able to get over this threshold. And we think that some of these single genetic factors, in fact, are very sort of impactful, have a very high risk associated with them. In contrast, what that may be telling us is that there are multiple genetic factors, each of one of which may not be as strong, that are accounting for more common, more commonly for individuals with diaphragmatic hernia. So one of the next things we did was to go on with the Gabriella Miller Kids First funding to be able to look at whole genome sequencing. That is to look at everything, not just the coding regions, but the non-coding regions as well. And as we did this, an interesting pattern started to emerge, not just because we could do whole genome sequencing, but because we could actually begin to look across congenital anomalies and start to see some patterns. And so we started this story with CDH, and people in my team thought I was dyslexic because the next thing we did is we looked at CHD, or congenital heart disease. And what we realized, and I put these in two columns here, one under diaphragm defects, the next one under heart defect, we saw, in fact, that we were seeing these de novo genetic changes in one particular gene, MYRF, in both individuals who had either who had CDH, and many of whom also had CHD. Interestingly enough, it was not just those two congenital anomalies, but you'll see in the column called genital defect. Some of them also had other genitourinary malformations. We had some of the babies who had ambiguous genitalia, some who had undescended testes as something that was milder, or in fact having no internal genitalia. We also saw that in some cases they had some other anomalies associated as well, so it looked like this was a gene that was having effect in multiple dimensions. I'll also show you that in the status column, unfortunately, many of these children didn't make it. Many of them were severely impacted, especially if they had both congenital diaphragmatic hernia and congenital heart disease. And so we were starting to also get some information in terms of prognosis. I'll also point out one unusual thing that has taught me a lot. Patients always teach me a lot, I have to say. We had one set, you may see it about 2/3 of the way down, called female identical twins. So we had one set of identical twins. Unfortunately, neither one of them made it. But even though they were genetically absolutely identical, and we've shown that with their sequence, one of the girls had a diaphragmatic hernia. The other twin had hypoplastic left heart syndrome, a type of congenital heart disease. And it just shows to me that even though they had the same genes, the same womb, the same in uterine environment, they both had certainly a manifestation of this, but not the same one. And so there are other factors beyond just the genes, even just the environment. And so what is this gene, MYRF? It's also a transcription factor, the theme for the day in terms of this. It was originally thought, and it was named after myelin, or a brain protein. It was thought to be mostly involved in terms of brain development, but I think we're just realizing that that was in some ways a misnomer, and I think if we had to rename it, we probably would name it differently because it seems to have a much more global effect in terms of body development overall. With the Gabriella Miller Kids First data, we're also going on to look much more systematically at the non-coding variation. So far, what we've seen is that we again see many new genetic changes within that region. We're starting to get a sense of what the impact of that is, but to say, I would say the short story is that it's complicated, and the impact of the things that we're seeing in the non-coding space is not as impactful as, at least per person or per genetic variant, as some of the variants we're seeing in the coding space. So there's more work to be done. So just to wrap up and say in summary that as we've done this, and just to give you a sense of this for any of the families who are listening in on this, this was really a whole community effort in terms of doing this. We literally have analyzed hundreds of families who have all worked together with us in doing this. And by doing this, we have started to chip away to understand that, importantly, something probably like about 15 to 20% of families now with CDH, we have, I think, a very good idea of what's going on that's causing their diaphragmatic hernia. I think we have an idea about some other genes that are contributing, and I won't have time to talk about it today, but we're also using this to be able to check this out and see if we're right by making mouse models of this to test our hypotheses. Most importantly, I just want to end by saying that we have a fabulous team driven by the families who really are, as I said, our partners within this. I want to put out a shout out to Julia Wynn and Rebecca Hernan, who are coordinators who've done a fabulous job with this. And just to say that it really does take a large team, fabulous team of dedicated surgeons, geneticists, neonatologists, gastroenterologists, but I'll stop here and be glad to take questions. And also I should acknowledge our funding, which has come from NIH as well as many of the families.
Wow, thank you, Wendy. I'm going to take back the controls just so I can show people. Wendy has to leave soon, so we're going to pause now to open the floor to questions for her. But please save other questions for the end of the webinar. So if you would like to, you can send me a chat. There's a little chat bubble, and you can message me that way. Or you can unmute yourself to ask Dr. Chung a question. I didn't share my screen yet. Well, I think everyone in the room with me is extremely impressed.
Yeah.
And it's so beautiful to see the story get told from the data. Any questions in here? Okay.
No, just this is very impressive.
Well, great. Thank you so much, Wendy. It was really great to see this story, and we hope to see, similar stories to come out of Kids First data in the future.
Very good. Thanks for having me.
Thank you. Okay, so now we will move on to the final, wow, we are right on time, which is incredible, to the final part of today, which is actually a overview and update of the program, and I will turn it over to Danielle Winchester.
Hi, my name is Danielle Winchester, and I am the NIH Commons Fund's Communication and Evaluation Liaison for the Kids First program. The last part of the webinar will focus on frequently asked questions we have received, including a brief overview of the Kids First and the different components of the program. One of the first questions we get, how did Kids First get started? The program was initiated in response to the 2014 Gabriella Miller Kids First Research Act. The act was signed into law just 5 years ago. The bill transferred $126 million that was in a presidential nominating convention fund to a Pediatric Research Initiative Fund and authorized the appropriation of $12.6 million per year to the NIH Commons Fund for 10 years. The first appropriation was in 2015. Gabriella Miller, which you have heard, the bill in the program's namesake, was a 10-year-old tenacious advocate for pediatric research who have lost her battle with DIPG just 6 months before the bill was signed. The vision of the program is to alleviate suffering from childhood cancer and structural birth defects by fostering collaborative research to uncover the etiology of these diseases and supporting data sharing within the pediatric research community. Collaborative research and data sharing are two key values that we feel will lead the way to achieving the ultimate goals of improving diagnosis and treatment for patients with these conditions. We are often asked, why is it important to study childhood cancer and structural birth defects together? The strength of studying these pediatric conditions together is supported by the fact that a risk factor for childhood cancer is being born with a birth defect, suggesting there are shared genetic pathways underlying these conditions. This graph shows a combination of specific cancers that are associated with specific birth defects. However, both sets of conditions tend to be relatively rare and therefore warrant coordinated efforts to combine, build, and study sufficiently powered data sets. This slide lays out who is involved in the Kids First program. Kids First is a NIH effort supported by the Common Fund. Each Common Fund program is overseen by an NIH working group. The Kids First working group is chaired by institute leaders from NICHD, NHLBI, NCI, and NHGRI, with representation from 11 other NIH institutes and centers, as well as the CDC. The Kids First program has implemented 2 major initiatives thus far. You have heard quite a bit about the DRC. Before the DRC was launched, we started the program with a large-scale data generation effort to identify childhood cancer and structural birth defects cohorts for whole genome sequencing by the Kids First funded sequencing centers. And in the next few slides, we will tell you more about each initiative. We have discussed and showed you features of the portal, but I would like to add that the Data Coordination Center and the Administration and Outreach Core are important components of the DRC. I mentioned previously, for our first initiative, we funded sequencing centers to perform the DNA sequencing of pediatric cohorts. For the last 3 years, we have worked with Hudson Alpha in collaboration with St. Jude and the Broad Institute. And in the first year, we worked with Baylor and WashU. Together, these sequencing centers have or are currently sequencing a total of 26,000 genomes. Now I will turn it over to Valerie, who will share with you some information about the cohort.
You probably noticed that the question, who is part of Kids First, is taking many slides to explain. So I'll tell you a little bit more about our XO1 cohort project. So in the first 4 years of the program, we have selected about 26 projects or cohorts, representing approximately 26,000 genomes, as Danielle described. That also represents an estimate of about 10,000 cases or patients for sequencing, and these are all at various stages of the pipeline. 7 of these data sets from these projects have already been released, which means that the data are available for analysis after approval, as you saw from Allison and Vincent's presentation. Each XO1 project is led by a team of investigators who provide samples from a cohort for sequencing, data sharing, and analysis. The cohorts or projects are roughly organized by condition, as you see here, but some projects study multiple conditions. For example, one project is made up of children who were born with a birth defect and then developed a childhood cancer later in life. So this project is made up of a variety of birth defects and childhood cancers. Some cohorts are also sequenced across multiple years, and this is because, based on our limited funds, we are often not able to sequence one large cohort in one given year. NIH program staff are thrilled with the diversity of conditions that continue to come in through our XO1 sequencing opportunities. It's enabling us to build a diverse and strong pediatric data set, and it also draws in a variety of expertise from these research teams. Last year, in fiscal year 2018, we were able to expand our sequencing efforts with support from other NIH programs. With the help from the Trans-NIH INCLUDE project, and INCLUDE stands for the investigation of co-occurring conditions across the lifespan to understand Down syndrome, we are collaboratively sequencing a cohort studying children with Down syndrome and congenital heart disease, or acute lymphoblastic leukemia, and another cohort of children with Down syndrome and myeloid malignancies. In collaboration with NIAAA, we are sequencing a cohort that aims to uncover the role of genetic susceptibility to fetal alcohol spectrum disorders. We truly feel that by working together, we can more rapidly foster genetic discovery, and we are really excited to actually demonstrate that with these projects. We have also recently established a collaboration with the Knockout Mouse Phenotyping Project, or COMP2, which is another Common Fund program. Although, as the name suggests, COMP typically produces knockout gene models, this collaboration is focused on COMP sites developing mouse strains to study phenotype and validate coding and non-coding genetic variants identified from Kids First datasets. So far, investigators have proposed variants from these 4 highlighted datasets, and you can learn more about these projects and the variant nomination process at the link that's on this slide. We are continuing to accept proposals on a rolling basis for investigators to nominate variants based on Kids First data until May 31st or until the funding for this pilot project are committed to projects. That said, we strongly encourage any researchers to collaborate with COMP to pursue collaborations even outside of this pilot. As you've seen, we are always looking for ways we can work together with other efforts in the spirit of accelerating pediatric research. But we also want to point out that within the program, the XO1 cohort investigators, sequencing centers, and the Data Resource Center also collaborate with each other, in some cases to address specific scientific questions or analyses within or across projects. And the impact of the program is amplified by the collaborations that each investigative team has already established. For example, several Kids First XO1 investigators are part of international collaborations to study specific pediatric conditions. Kids First sequencing centers are often involved with other large-scale data efforts to inform other Kids First projects, and the Data Resource Center is charged with facilitating new collaborations as well as engaging with patients, families, and foundations. So each component of the program is designed to draw in more researchers and expertise as we continue to build a Kids First research community. But of course, we also consider any researcher who makes use of or makes discoveries with Kids First data to be part of this community, which leads to our next frequently asked question, which is how do I access data? As you saw from today's demo, anyone can register and log into the portal to search, filter, or visualize information about Kids First datasets. But in order to access individual-level sequence data, investigators must submit a data access request through dbGaP, which will be processed by our Kids First Data Access Committee. After approval, data sets can be accessed and analyzed through the cloud-based workspaces that are integrated with the Kids First Data Resource Portal. Last September, we hosted a day-long webinar with the Data Resource Center, and one of these presentations went over lots of detail about how to submit a data access request. So we actually encourage you to watch that separately, and there are also other webinars from that day that may be of interest to you. And now I will turn it over to James Coulomb.
So a question that we've been asked is how does Kids First support data sharing? And first, I want to stress that as part of stimulating and accelerating pediatric research, we consider data sharing to be extremely important, and we've taken several steps to support data sharing. First, when selecting cohorts for sequencing, a priority for us are cohorts whose data can be shared and analyzed in combination with other data from other conditions, and because all data sets are not consented that way. We created and have posted FAQs to explain the genomic data sharing process and clarify our expectations for data sharing in the Kids First program.
Kids First frequently asked questions are available at commonfund.nih.gov/kidsfirst.
To speed up access to Kids First data, we formed a separate Kids First Data Access Committee to streamline these data access requests. We are also discussing with other NIH programs ways to facilitate data access across programs. And of course, as you have seen, we've funded the Data Resource Center to build an internet portal and some very impressive tools to facilitate data analysis and analysis across data sets. Oops, what did I do? Push that. There. Okay. Another question that comes up is how are Kids First data sets selected? First, researchers apply to have their cohort samples sequenced by the Kids First sequencing centers through the X01 mechanism. These X01 proposals are discussed and scored by a scientific peer review panel, and then the Kids First working group makes selections based on a number of criteria. We consider the scientific and technical merit based on the peer review scores and written critiques from the review panel. We consider broad data sharing and ability to combine data across conditions, as was mentioned before, as a very important factor in our selection process. And we try to evaluate the perceived overall data value of that data set for the Kids First data resource and try to balance our investments in childhood cancer and birth defects. We also consider the availability of the samples and the quality of the samples and their suitability for sequencing. Okay, a frequent question is what funding opportunities are available to support Kids First research? You can apply to have samples sequenced through the X01 process, and the funding opportunity announcement shown here is out of date, but we expect to reissue this announcement for the next 2 years, although this announcement is a good guide for preparing those applications so you can look ahead to see what we're asking for. You can also apply through the Kids First R03 announcement for analysis support or through other mechanisms such as the NIH parent R01. In order to find out about future Kids First funding opportunities, I strongly suggest that you sign up for the Kids First listserv at the address shown here.
Join the Kids First listserv at commonfund.nih.gov/kidsfirst/register.
Yep.
James discussed how funding opportunities that are available for the Kids First program, but for community members, healthcare professionals, patients, and family members, if you would like to get involved with the Kids First program, you can search data that's available through the Kids First Data Resource Portal. which is at portal.kidsfirstdrc.org. You can also connect with and provide feedback to the DRC by sending an email to support@kidsfirstdrc.org. You can contact the program for questions directly or feedback by sending an email to kidsfirst@od.nih.gov. For additional information about the Kids First projects, you can visit the Kids First X01 project page and read each abstract. If you want to learn more about the Kids First investigators, we encourage you to view their profiles on the Kids First DRC website. For more information about available data sets that Valerie discussed, you can visit the Kids First DRC support page. And lastly, these are helpful links for you to visit after our presentation. The first one is the Kids First program website, which is commonfund.nih.gov/kidsfirst. The second is the Kids First DRC website, which is kidsfirstdrc.org/. And lastly, you have the portal website, which is portal.kidsfirstdrc.org.
Okay, and now we will open the floor to questions. If you have questions for us, program staff, we're here in the room, or if you have any questions for the Kids First Data Resource Center team. I believe Wendy had to leave, but you can always email her or email me questions, email the Kids First mailbox questions, and we can get those to her if you have questions about her presentation as well. I saw a couple comments come through the chat. Very encouraging. Thank yous, basically, but no questions yet. I'm not hearing anything, and I'm not receiving any chats yet. We'll give it another minute. I'm sure you're all feeling just as overwhelmed as we are with the amount of information that just came at you.
On the DRC side, I just wanted to reiterate what Valerie mentioned, which is once any users interact with the portal environment or the resources we provide, you have several opportunities to both inform development, but also feel free to use the support email to ask any questions on the usability of the portal or any specific questions regarding access, analyses, or opportunities to leverage the workspace environment as you explore.
Thank you, Adam. We got a comment that this was a really useful webinar, which is what we really like to hear. Okay, I think we'll go ahead and close early so that everybody can go have lunch, or depending on your time zone, I suppose. So again, we will make this webinar available on the Kids First Common Fund website. It'll take a little bit of time because we have to make it 508 compliant. But in the meantime, feel free to contact us, and we could email you a temporary WebEx link to watch the webinar. Oh, there is a question. Here we go. I have a question about the RO3 analysis proposals. The funding announcement says to contact the Data Resource Center to coordinate proposals. Who's the point of contact? I'll emphasize that you can contact the Data Resource Center to coordinate with them on the RO3s. And yes, so it would be the support@kidsfirstdrc.org email address.
Yeah. And our goal as a DRC is to really vet and provide, a complete picture of the resources that might be available within the DRC to support your analysis and proposals, and really partner in providing such resources and their availability.
That's right. We don't want you to build a tool that's already available through Chavatica, essentially. Okay, great. Thank you, everyone. Feel free to contact with any questions ever.